neurotransmitters alter the resting membrane potential in the same way that potassium and sodium would? Like, would both groups alter the same thing, the resting membrane potential?
They could, but they don't have to. So the question is, ionotropic, metabotropic neurotransmitters, do they alter the membrane potential? One's obviously direct and the other one's indirect, but do they ultimately alter it using the sodium and potassium voltage-gated channels? No, they don't have to. Okay? So you'll see later in the semester, for example, when we look at a process that's found in the inner ear, we actually see, instead of sodium coming into the cell, we see potassium coming into the cell. But you could actually think of an organism that used calcium or any other positively charged or negative. You could use chloride. You could build an alien model, right? And you could say, physiologically, we're not going to bother with positively charged ions. We're going to move negatively charged ions. So it doesn't have to be sodium and potassium. It just happens to be that for us. Now, sodium makes a lot of sense to have high outside the cell, right? Because sodium likes water, right? Sodium likes chloride. And so you'll have a fluid environment that has a saline solution. And that's very, very hospitable for life. And so that's one of the reasons biologically that we believe we have sodium high outside the cell. The salinity is just right.
I think in the first lecture you said that the myelinated sheath is more energy positive, right?
Yeah.
So how come?
To build initially.
Oh, okay, so not when the...
Yeah, so the question is, okay, I'm sorry, go ahead, finish your question.
Well, not when the electricity's going through it, right?
Not when, I'm talking about when the organism is being built. It takes more biologic energy to wrap a neuron in myelin than not to. But then you reap the benefits later because those myelinated axons are more efficient and faster to use. But it was at the expense of biological creative energy when the embryo was being developed. Okay? So if you look at the brain, I think I was saying this, when you look at the brain, you slice through the sheep brain, you can clearly see gray matter, which is unmyelinated. You can see white matter, which is myelinated. The whole brain isn't myelinated. So we pick and choose, or create, the mechanisms that are in place to build a human have decided that these are critical areas to myelinate and these ones are not.
Later on in life, does the brain get myelinated, or just like
The nervous system, the central nervous system, is one of the last things to develop completely. So in humans. So that's why we don't come out walking and talking, and infants can't even see very well at all. And then they put like this goop in their eye and they're like, right? So the nervous system is one of the ones, that's the whole reason in pediatric medicine they want you to do like tummy time, and they want to have the mobiles and playing around. And they've done some pretty crazy studies in primates where they restrict a lot of that developmental influence, and the nervous system doesn't really develop. Okay, so we do know for a fact that the nervous system is one of the last systems to develop. Okay. Other questions? All right, so this is lecture 3. We're going to get as far as we can today, as time will allow. You'll see in the very beginning, though, that it is a lot of review. Okay, and that's intentional. Because the heavy stuff is really stuff we've already covered. Now I'm just reinforcing the mechanisms. But we have a whole lecture, we'll spill into Monday if we need it. We will. To reinforce all of these important mechanisms and stay on track. Alright, so let's talk here. After being released, a neurotransmitter binds to receptors where? A, postsynaptic membrane, neuromodulators, synaptic cleft, synaptic vesicle. How many of you say A? The A's would be correct. Neurotransmitter binds to the postsynaptic membrane. Okay? It doesn't bind in the cleft. That's the gap. Okay? Alright, how about this one? These are possible test questions. Neurotransmitter attaches to its postsynaptic receptor. And this eventually induces the production of cyclic A&P. So this neurotransmitter is most likely ionotropic, metabotropic, inducing a change in charge across the membrane, 2 of the above or none of these. How many of you say A? Okay, good. How many of you say B? How many of you say C? How many of you say D and E? Why did you guys say D? I want to hear the answers. Yes?
Because the metabotropic neurotransmitter does, in a roundabout way, change the resting membrane potential. So
I would
both
agree.
happen.
So I would argue that if this was a test question, it would be shame on Keller, a bad test question. Because there would be 2 possible answers. There would be B and D. And if you looked at your exam and you put D, with that explanation, I think you could argue those points back. Okay? So metabotropic and D, 2 of these. So it would be B and C would be the 2 correct answers.
What would your answer have been done?
I would have chose metabotropic because it eventually does a change across the membrane, but it first uses a second messenger system that eventually leads to it. But I agree with your rationale. I would say they're both correct.
I don't get why it's a weird question because D says 2 of these, and if B and C are both accurate.
I'm saying that I would have said the correct answer was B.
Oh.
But you guys are arguing that it's B and C, and I'm agreeing with you. What's that?
So you mean the same?
No, this is an example of a test question so that you guys can see how you need to read them and how you would argue back points. Okay? You guys with me? So how can I change the question to be more accurate so B is the only correct answer? Get rid of option D. That would be easy. Okay. Well, actually, C would probably be better. Change C to something else. Okay? That relates to ionotropic. Change C to like the production of this neurotransmitter is acetylcholine. If I changed C to acetylcholine, then there would only be 1 correct answer. You with me? All right. Take out a piece of paper, or on your notes, I want you to draw an action potential. What's that? Yeah, draw an action potential, like the graph. After you've done drawing it, label these components. This is something else you'll be doing in SI. Practicing drawing the action potential.
Keller,
Yes?
on our exam, could you give us that diagram? We would maybe have to identify what the number is as one of these.
I could do that, yeah. On the exam, I could give you a diagram and maybe ask you to identify points on the diagram. That's very reasonable question and a good way to forecast what possibly could be on the exam. That's kind of why we're doing this next activity. Okay, how many of you are still drawing? Okay, I'll wait, that's fine. It depends, but usually much longer than the action potential. Okay, ready? Does your drawing look something like this? Okay, good, because you're like, yeah, because I printed out the notes and so I just copied the next slide. Yeah, you guys are brilliant, I know. All right, so here we got our action potential. Let's go over a few of these. So this is just a different way of seeing the same information that we've been looking at for two and a half lectures now. So do you think that action potentials are good to know for the exam? I hope so. Should you be familiar with the different components, as was asked? Yes. Okay. If you look here and analyze it, this is minus 70, right? This is our millivolts. You can see our positive is here. Here's our threshold at minus 55. You can see relative time in milliseconds. And this is the point at which you've got these little postsynaptic potentials. That means it's a potential, a graded potential, or a local potential that never reached threshold. So we call them failed initiations. Right? So they fire, but they don't ever cross threshold, so they don't do anything. But this one right here actually reached threshold. Now you get depolarization. This is considered an action potential. Now there's no turning back. It will fire. You can't reverse it. You can't stop it.
Is the action potential the moment where it does fire?
The action potential is referring to this whole thing, including repolarization. Then you have repolarization that comes down on this side, and you return past resting membrane potential. This is drawn very exaggerated to prove a point, but the refractory period goes way below, and then it returns back to resting state. And at the very end, the sodium-potassium pump that's constantly moving helps to reestablish and maintain that resting membrane potential. Okay? Question here.
Realistically, and kind of as your body actually works, do you ever really have just a resting state, or is there always local?
Yeah, that's a great question.
Happening?
So throughout the peak of your day, the question is a good one. So realistically, under normal activity, do your cells ever really stay at minus 70 at resting membrane potential? Or are they constantly seeing local potentials? And the answer is the latter of the two. You're constantly receiving input throughout your day. There are sounds that are happening. People are talking. Things are going on that you don't even notice. Right? Some of it reaches a threshold and you pay attention. So throughout our day to interact with our environment, you're getting these local potentials. It's when they reach a threshold that grabs an action potential that anything ever really happens. And that's one of the reasons I was in this conversation with some faculty just last week about the nervous system. And there's some new diseases that are being discovered. And some of these diseases are related to insomnia. And patients not being able to sleep or not being able to sleep well. So one of the things that we know that's well documented, during the day, our neurons actually retract away from each other slightly. So those synaptic clefts get a little bit bigger. And then in the evening when you sleep, they actually come back towards each other. So this process, it's not really nerve regeneration. It's just the plasticity of the nervous system. As you fatigue throughout the day, your body does need rest. And we've always asked ourselves in the field, why do we sleep? What's the whole point? Why is it so beneficial? Okay? And this came on the heels of a talk that was given, a great talk that was given last week on hibernators. This is totally off tangent, but today looks nothing normal, so that's okay. These are hibernators. These are mammals that actually hibernate. They'll get their core body temperature to below Celsius, 0 degrees. So they'll be like minus 4 Celsius. That's their core body, and they're hibernating. Well, when they hibernate, they're actually not asleep. Yeah. So they actually have to warm their core temperature up to get to a sleep state, because sleep is a certain theta rhythm that's measured in the brain. And so in a winter season in Alaska, these mammals will actually, you'll see them in the summertime, their body temperature's up here. Then in the wintertime, it drops to minus 4. Every 12 days it comes up, and then it comes back down. Every 12 days, throughout the entire winter. And they're coming up to go to sleep from a hibernating state. So it gives, just a second, so it gives credence to this concept of sleep. So the aspect as it relates to this lecture, why am I talking about that, is the plasticity of these nervous tissue and these neurons is so critical, and they're used all day long, especially if you're engaging with your environment, that when you go to sleep at night is when your body has the ability to kind of reset everything. Question?
Then what are the hibernating animals doing when their core body temperature is at -4 centigrade? What are they doing if they're not technically sleeping? Are they like close to being dead, or?
I don't know. I have no clue. Some of the theories, it's a good question as to why do they need to raise their temperature to sleep. What causes that? Again, this won't be on the exam, but the buildup of certain metabolites, maybe.
Yeah.
Like if you're hibernating at -4 degrees Celsius, can you actually get rid of your urea?
No.
No. So maybe they warm up to take a leak, and then they hibernate again. Okay. Okay. No more questions. We're going on here. I'm cutting everybody off. Cutting you all off. I want you to take your graph, and I want you to label it with these 7 numbers. 1, 2, 3, 4, 5, 6, 7. I have office hours today if you have more questions. So you can come see me. We can talk about how hibernators poop and pee and anything you want to. Everybody got 1, 2, 3, 4, 5, 6, 7? Okay. Write down the number which tells you where almost all the voltage-sensitive potassium gates are closed. We just went over this like 15 minutes ago, so hopefully this isn't too complicated. Did you guys get that? Who has an answer? And who has an explanation?
Number 6?
Number 6. Why?
I think so because at 3, 4, and 5, the potassium gates are open to allow potassium to rush or leak out of the cell. And when it's too much has left the cell at number 6 during hyperpolarization, they close to restore the resting membrane protection.
Perfect. Did you guys follow all that? Yeah. Can you hear it in the back? Okay, good. You're all nodding yes or falling asleep viciously. Okay. Write down the number which best represents voltage-sensitive sodium gates are closed and inactive. And inactive. Closed and inactive. Could there be multiple answers? I would say yes. Is this a mess up on my part? No. This is intentional to show you how to interpret the graph. So, who has an answer and who can defend it? Sodium gates are closed. So, I'd love a new participant today. It would be great. Yes, sir. Right there, please.
2 and 4.
2 and 4. They're closed at 2 and they're closed at 4. Why do you? I agree with 4. Why do you say 2?
2. My understanding with 2 was once the stimuli reaches thresholds, then the voltage sensitive gates open. That allows the sodium.
Yeah. So right at 2 is where they're going to open.
Oh,
But I would. How about I substitute 1 for you?
okay.
Because in here they're still closed. So I would say 1 and 4. Anybody have any other answers? 5, they're still closed. Any other answers? 6 and 7. All of those would be correct. The only ones that wouldn't work are actually probably 2 and 3. Because that's when they're open. Okay? All right, let's try this one. So this might be a way that I'd ask the question on the exam. You guys catch that part? Okay. Write down the number which best represents all the voltage-sensitive potassium gates are finally open. Like, I don't like these finallys and near close. I'm trying to be intentionally vague so that I can pull out multiple answers from you. I won't be vague on the exam. There will only be 1 right answer. Or 2, and then it'll be A and B only, right? All right, what's this one? Who's got it? All voltage-sensitive potassium gates are finally open. Way in the back, yes? Okay. starting to repolarize. In which direction would potassium be going at 5? Into or out of the cell? Out. Very good. Very good. Anybody else have an answer? Other than 5?
Maybe 6.
Well, 6, they're almost closed. 4, I would agree. They're open. They're starting to open at 3. You'd have a weak argument at 3. I would say the better way to describe 3 would be they're starting to open. 4, they're open. 5, they're open. 6, they're closing, but they're still open. Okay? How about this one? Ooh, this one has a 2-part. Voltage-sensitive sodium gates are open, and voltage-sensitive potassium gates are nearly open. I like 3 the best. 4 is where the sodium gates are closed. 2, the sodium gates are open, but the potassium gates are not opening until 3. So I would say that 3 is the best answer. Point at which the process is still reversible. Still reversible. 1. Why? Haven't hit threshold. Okay? It's considered a local potential. It's considered a what? What's another name for local? Graded potential. Okay, all of those would be correct reasonings behind number 1. How about this one? Write down the number that best represents the cells at resting membrane potential, but ions may not yet be in their proper location. So firing may not be possible. 6, why? Too much potassium outside the cell. It's the refractory period. All of those are correct explanations. You guys, sounds like you're getting this. Okay? Does this make sense? Let me show you another picture from a different textbook, because here's what notoriously is difficult for students. Is you learn something, and then I change the color of the stinking potassium channel, and you're like, what's up, dude? I mean, okay? I can't, is it blue or is it orange? I mean, it's like black and white, man. I don't know. So on the exam, how many of you had a color exam in living color? No, in my class. We're too cheap in this department, so. No color exam. So don't memorize the colors. Okay? If you see a channel, you're going to need more than just like, well, I don't know. Could you tell me, Dr. Keller, if this is a blue channel or an orange channel? So don't study the colors. Okay, so let's look at this one. Okay, this is the same stuff from a different textbook intentionally to encourage you to understand the concept and not memorize figures and colors and answers. Okay? So if we start down here at resting state, and we're going to move 2, 3, right, 4, and then we're going to go to 5. We're like, well, what happened to 6 and this is a different textbook. There's no 6 and 7 here. Okay, so again, if it's numbered A, B, C, you know, like is that 1 or is that 6? Okay, if I use the alphabet instead. So here's our cytosol. Here's our extracellular fluid. Here's our sodium channel that's closed, right? Here's our activating gates that are being shown. And here's our extracellular fluid out here. Here's depolarization. In depolarization, sodium channel is open at 2. Here's where 2 is stimulated, right? And so some sodium is coming in. Hasn't reached threshold yet, but when it reaches threshold, now you're in the 3 phase. So all sodium gates are open. Potassium is being shown as being closed at 3. We just said in a different textbook that these start to open here. So you could see the differences between when you read one book versus another. Okay, these slight little differences. Then you go over here to 4. This is on this side. This is the falling phase. So our sodium gates are both closed. There's no difference, no discrepancy between the textbooks there. and our potassium channels are open, and so that brings this down. Here you can see that these potassium channels are still open, and you're still losing some potassium, but the sodium gates are still closed at 5, and that's our undershoot, as this textbook calls it, instead of hyperpolarization or refractory period. So just a different take on the same information. Okay? Question? Yeah, so this is saying an undershoot, meaning that this is shooting down. Overshoot is saying that it over-depolarizes, or over-polarizes, so it's hyperpolarized. Okay? All right, we're going to stop here, and we're going to pick up at the stopping point on Monday. How do you stop a nerve signal? Okay? I'll see you in my office hours. They're 4 to 5 today. Or please go to SI.
Are you giving back today during your office hours?
I can if you would like.